Hi everyone, my topic is phenolic composition of grapes in relation to yield control practices in vineyards. As we all know, phenolics play an important role in pigmentation, growth, and sensory properties of berries and wines. It's one of the indicators of grape quality. In wines, phenolics are responsible for color, mouthfeel, and taste. Anthocyanins are the main phenolic compounds in red grapes, whereas flavin-3-ols are more abundant in white varieties. Each cultivar presents different phenolic composition in berries. At the same time, concentration and distribution of different phenolics are significantly influenced by other factors. In this video, I'm going to present the change of phenolic composition in grape berries during ripening and the effects of yield control techniques in vineyards on the phenolic content and distribution of grapes. First, let's talk about the evolution of phenolic during ripening. This is from a research concerning phenolics in relation to vineyard nitrogen and potassium fertilization. We can see several different stages in the development of total polyphenol contents in grape skins. First, there is a relatively slow and continuous increase during the first stage of ripening. Then we got a sharp accumulation during the 5th week after horizon. Then a really short lag phase and a final reduction until harvest. As an important pigment, skin anthocyanins usually increase beginning with horizon until reaching the maximum value and then diminish when the berries are overripe. And tannins in skin are synthesized in the early stage of berry development and change relatively little from horizon to harvest. They also found that total hydroxycinnamates show the maximum levels prior to horizon and then a decline in the later stages. The quercetin glucosides were highly accumulated in berries during early stages and showed a sharp decline after horizon because its precursors are used to produce anthocyanins. Now I'm going to talk about how yield control practices impact phenolic compounds in grapes, including leaf removal, cluster thinning, and some combination of different techniques. First, leaf removal is one of the practices that can control yield in vineyards. There is evidence that leaf removal increases the concentration of phenolic compounds. The authors used vines of Pinot Noir grown in the Pava Valley to investigate the effect of leaf removal timing. They removed the basal 4 to 6 leaves of all shoots at berry set and at horizon. So they got different light exposure and different temperatures on the berry surface depending on timing of leaf removal. The large white circles in this chart represent grape berries, and black circles represent amounts of anthocyanins and flavonols. The results show that leaf removal significantly increased the concentration of anthocyanins, both at berry set and at horizon, but the increase was more significant in the berry set. Flavonols were more affected by the leaf removal practice. Hydroxycinnamic acids were affected slightly in this research, which means at berry set and at horizon. But leaf removal before blooming significantly lowered the hydroxycinnamic and hydroxybenzoic content in another research. There are other papers studying leaf removal before the blooming. This time, they removed at 3 different stages, before blooming, at berry set, and at horizon. This time, the concentration of all the hydroxycinnamic and hydroxybenzoic are significantly lower compared with other treatments. In addition, 75 to 80% of leaf removal before blooming help wines extracted more anthocyanins, so that wines have better color intensity. By the way, I used orange to indicate an increase or higher content, and blue means a decrease. Thus we can see the removal leaf practice will influence phenolic compounds in grapes, but the increase or decrease effect depends on timing of removal and the type of phenolic compounds. Cluster thinning also has the effect of increasing phenolic compounds. In this study, clusters were removed 1 month after blooming. The concentrations of anthocyanins and flavonols in berry skins were significantly increased. In another research conducted in China, 5 treatments were studied to investigate the effect of cluster thinning. Distal clusters were removed at blooming and at horizon. Also, basal clusters were removed at blooming and horizon. No cluster removal is a control group. The results indicated that the concentrations of catechin, epicatechin, and epigallocatechin in each of the removal treatment groups were larger than that in control group, especially when the distal clusters were removed. These 2 studies illustrate that cluster thinning really can really promote berry development. However, the effect of cluster thinning on phenolic compounds also might differ based on other factors. Another person called Fenzon studied the effect of cluster thinning timing on the phenolic composition of Malbec skins and seeds. 50% clusters were removed at 40 days, 80 days, and 100 days after blooming. The results indicated that the effects were different depending on vintage and also depending on the tissue. For example, in 2008, cluster removal at 40 days after blooming increased biosynthesis of individual anthocyanins in skins, whereas in seeds it mainly changed the concentrations of other chemicals. While in 2009 it significantly affected the content of flavonols in skins, whereas in seeds it modified different chemicals again. Sometimes grape growers will combine several methods together to manage canopy. However, the combination of different canopy management could not guarantee the berry quality improvement. In a study on Cabernet Sauvignon and Sauvignon Blanc grown in India, there are 3 practices combined together, which are shoot thinning, cluster thinning, and leaf removal. It turned out that the combination of leaf removal, either with shoot thinning or cluster thinning, showed the highest anthocyanin content, while the vines with 3 treatments together showed the lowest anthocyanin concentration. So this study indicates that excessive light exposure is not appropriate for grape wines. This experiment showed how trimming and cluster thinning influence phenolic compounds. Wines were applied 0, 50, and 75% of cluster removal and shoot thinning treatments. In this chart, cluster thinning with different letters in columns means significant difference. Trimmed and untrimmed treatments in each cluster thinning with a star are significantly different. For Merlot, no cluster removal treatment showed higher anthocyanins and total phenolic compounds in the skins, but lower total phenolic contents in the seeds of untrimmed vines. In 50% and 75% cluster removal treatments, the trimming treatment increased anthocyanins and total phenol levels. It shows that shoot trimming treatment did not increase skin phenols and anthocyanin contents of Merlot without cluster removal treatment. For Cabernet Sauvignon, there was no significant effect of trimming on anthocyanins and phenols. The cluster reduction increased the level of these phenolic compounds. In both cultivars, the anthocyanins and the total phenolics extracted from berry skins were more affected by cluster thinning than shoot trimming. The relationship between phenolic compounds and crop yield in this study was not really clear, which could be caused by different cultivars and interaction of different practices. Finally, there is a conclusion. The phenolic composition of grapes can be affected by many factors. Varieties, environment, such as sunlight, rainfall, and soil condition, viticultural practices such as irrigation strategy, yield balance, vine training, and use of fertilizers. All these factors are potentially or strictly correlated with each other. Also, based on the studies I mentioned in this video, the viticultural practices can interact with each other, enhancing or reducing their effects. So remember, we need to consider many factors when we want to manage our vineyards with viticultural methods. Any treatments or practices cannot be considered individually. There are some references and studies I mentioned in this video. Thanks for watching and hope you enjoyed it.